Nope, it's actually postponed.
Did they postpone it?
Yeah, they just did.
This one was like, nope, we're going.
Well, I'm pretty sure Morris was the meet where they laid down plywood to get to the pits, to get across that little ditch in the. So it would not have surprised me if we had one, honestly.
I was thinking that we would have it. You guys don't have it? No.
I think we're still gonna have practice though. So.
That is the worst when you bring all your stuff to like a game or a meet and it gets canceled, but they're like, oh, you still got practice. But you don't have practice stuff. Yeah, and all you have.
But we get out at 1 o'clock today, so they can't really complain.
Well,
So.
I just have my sweatpants and a sweatshirt because I wasn't planning on it.
Right. Yeah.
And I
Yeah,
came
yeah. Okay. So we finished up on amphibians and how they're still tied to the environment and all that. So let's just do some review questions and we'll move on to reptiles and how, amphibians, they were kind of like the first draft of animals that could live on land, and now we're going to try to improve on some of those features. So select the feature that does not distinguish living amphibians.
B.
Yes, it is shoulder muscles, correct. The Ithacostega represents.
I know it's like a fossil, but
Mm-hmm.
so C.
C, the first amphibian, yeah. Why was the evolution of the pulmonary veins important for amphibians? A, to move oxygen to and from the lungs. So this is where we actually have lungs. All right, moving on to reptiles. So I'm going to talk about 3 kind of big groups about reptiles, or 3 orders of reptiles. Distinguish between synapsids and diopsids. That's actually kind of a fascinating topic itself. And then the big thing about reptiles is this development of something called an amniotic egg. We're cutting ties with water with an amniotic egg. Whereas with frogs, when they laid their eggs, it had to be in a moist environment. Here, we have the ability to not dry out or desiccate. So amphibians were the first draft for survival on land. And reptiles just kind of made some innovations, some improved innovations of it. Okay, so one of the improved innovations is their arrangement of legs. So if you remember back with amphibians, yeah, they had 4 limbs, 4 limbs, or 4 limbs and hind limbs, but the way that their hind limbs were arranged, their bodies still rested on the ground. So now with reptiles and their 4 limbs that they have, their body is off on the ground. Okay, so they can actually lift their entire body and keep it off the ground, whereas with frogs, they kind of sit like a, I don't know, my son when he's, sitting on his butt and his legs, and he's got his, hands out. Lungs and heart are more efficient. We're gonna see that we're gonna develop more chambers. Does anyone remember how many chambers of heart or chambers are in the heart of an amphibian?
2?
That was fish.
3.
3. And now with reptiles, you're gonna see 4. Okay, so it's gonna be way more efficient. The lungs, we're gonna see increased surface area with lungs. The skin, it's not going to be moist and smooth like amphibians. In fact, it's going to be made up of dry plates, scales to help minimize water loss. And then I'm going to talk a little bit about the amniotic egg on this next slide. Watertight coverings for the eggs. So 3 key characteristics for reptiles. First one is this amniotic egg. So all reptiles, birds, and mammals pretty much show the same pattern of membranes within their egg, and we're actually called amniotes. So we're kind of included in this this characteristic, but obviously our egg is a little bit different. Okay, so the amniotic egg has a food source, the yolk. So if you just think about, the eggs you buy from a store, that yolk is a food source for the developing embryo on the inside. The amniotic egg has 4 membranes, and so I think I listed it in order, kind of like from the outside to the inside. But the outermost membrane of the egg is called the chorion. It lies just below the shell, and the shell is porous, so gas exchange can happen between the shell and the inside. But it doesn't allow water to, so it retains the water. So we can still have gases going through, but not water. Then we have the amnion, which is where the embryo is encased in, and it's just a fluid-filled cavity. We have a yolk sac, so it provides food for this embryo through blood vessels. And then finally the allantois, which surrounds the cavity, and this is where waste products are excreted from the embryo. Okay, so this next slide, you don't need to write anything. It's a slide that I got from my master's course when I was learning about reptiles in the amniotic egg, and I just thought it was a really nice slide, nice diagram. So here we have our outermost shell, okay, and then the chorion is here. Okay, so it'd be this layer. And then we have the amnion, which is not, oh, yep, there it is, right there. Okay, and then the allantois. There's the blood vessels, stores nitrogenous waste, and then the yolk sac is basically like all of this. But this is the diagram that your book kind of gives you. So there's the embryo, and you can see that it's tied to blood vessels for the yolk sac for the food. Okay, a second key characteristic is dry skin. So amphibians were moist and smooth, and reptiles are going to be dry. I'm not going to say bumpy because there are some reptiles that can be smooth. But it's dry, it's watertight skin, and that skin is made up of scales. These scales are made up of keratin. It's the same protein that's used for claws, fingernails, and bird feathers. You can see over here a cross-section of it, and the dermis is kind of made up of 2 layers, if you think of it. The uppermost layer is loosely packed cells, and you kind of see that right here. And then the deeper layer is more densely packed. And the dermis is where, you're going to have muscles, nerve fibers, chromatophores for pigments, adipose tissue, just fat. The coloration of some species is obviously a function of chromatophores, which we mentioned with the octopus. They can be quite elaborate. There are some organisms that can change colors, like chameleons. Okay, so it's kind of fascinating how they can change very quickly in the response to their environmental stimuli. But the coloration from these chromatophores, they can have lots of functions, like when it's mating season, thermoregulation, sometimes social status, especially with lizards and their little like beard that they have. So, yeah. What else? I feel like I was going to say something else. Oh, yeah, these pictures. Okay. So, another function for the scale, the skin, sometimes we see glands and sometimes we don't. But if they do have glands, the smells that they can emit can be pretty nasty. But obviously, they produce these strong smelling substances to attract mates or to warn predators to say, hey, get the heck away from me. They also don't have any sweat glands, so they can't sweat to cool themselves off. So, like we'll talk about how reptiles are ectotherms. So that, to warm up, they might sit on rocks to heat their body temperature. And then, like, to cool off, obviously, they got to move off the rock and find a shaded area to bring their body temperature back down. But the scales can also be used for climbing. We were going to do a study on geckos. Did I ever make you read a scientific article about geckos? I think I had to throw it out because the website didn't work anymore because we lost, what's the word I'm looking for? Flash. And it didn't work anymore. So, okay. Third key characteristic for reptiles is thoracic breathing. So amphibians, they are limited how much they can take in because their lungs are kind of small. But how they took in is that, or took in gases or air, is they squeezed their throat to force it into their lungs. So how big their mouth was as much air as they could get at one time. But with reptiles, now we're going to develop something called pulmonary breathing, a rib cage that surrounds the lungs that's going to expand and contract and suck air in and force it back out. And so now how much air we can take in is limited by how big your lungs are. Okay, so frogs, like I said, they would have to open their mouth and force air in, and then they would close their mouth, and then they would take the air that's in their mouth and force it into their lungs, and then that's how they do breathing. Whereas now with reptiles, we're going to have muscles and a rib cage and a diaphragm in some cases to help expand our lungs and contract them, or expand the lungs and contract them. So that's just extra info. Okay. So reptiles, they've been around for a really long time, 250 million years. We've seen a series of different reptiles appear, and some of them have disappeared. But we can basically classify these amniotes into three major lineages: the Anapsids, the Synapsids, and the Diopsides. And we classify them according to the holes that we can find in their skulls, specifically holes that are behind their orbital hole. I feel like that's not the right terminology for that. The socket, orbital socket? I don't know. So here we have a skull, the anapsid, and that's the orbital socket, and there are no holes behind it. So anapsid, no holes. Synapsids, they have one hole behind the eye socket. And synapsids are going to be responsible for the mammals. Eventually they're going to give rise to the mammals. And then we have the diopsids, where we have two holes behind the orbital socket, and it's going to give rise to other reptilian groups. This also includes the birds. So yeah, the presence and the number of openings behind your eyes is how they are all grouped here. You may be wondering, what's the purpose of these holes? Well, these holes serve as attachment points for jaw muscles. And so jaw muscles can anchor to these holes, and as a result, we can get some pretty powerful bites. So yeah. So here's a fossil of Tyrannosaurus rex, and you can see that we have one big fossil. Oh, you know what? Yeah, never mind. This must be the orbital socket, and then these are the two diopsids. Okay. So this is the picture from your book that talks about the anapsid and the synapsids and the diopsid skull structures. So we'll talk a little bit about each one here. Synapsids. They were probably the earliest of the reptiles, and they include something called the Plachiosaurus. They dominated for about 50 million years, and then they kind of died out. But at that time, 250 million years ago, 70% of all land vertebrates belonged to this group. Their teeth were long and sharp, and they described them as steak knife teeth. So it enabled them to kill prey their own size. The Therapsids dominated for about 20 million years, so around 230 million years ago. There is some evidence with fossils that they were endotherms, which means they produced their own body heat. They didn't have to sit on rocks, bask in the sun, all that. There are indicators that maybe they possessed hair. They were replaced by the diopsids, which I'm going to discuss on the next slide, but one group survived and has living descendants today, which are mammals. So this is some artist rendition of what possibly that therapsid early organism looked like. Okay, diopsids. One group of diopsids is called the Archaeosaurus. They were ectoderms, like our amphibians and early reptiles. And this is going to be the group that gave rise to crocodiles, which I'll discuss later on in this section. Pterosaurs, dinosaurs, and the birds. This group is responsible for some of the largest animals that the world has ever seen, aka dinosaurs. Some were bipedal, which means they're placing their weight directly over their legs, and as a result, they got great speed and agility from this posture. And you know, dinosaurs, they ruled for 150 million years until, until an asteroid or a comet, whatever it was, struck Earth and caused a mass extinction 65 million years ago. So who knows where mammals would be if they were not wiped out. All right, important characteristics of our modern reptiles. We have internal fertilization, and the purpose of that is, okay, reptiles, they have this leathery egg, and if that was, laid, there's no way the sperm could penetrate through all those membrane barriers. So instead, what we have to do is get the sperm inside the female and fertilize the egg before those protective barriers form. So we have internal fertilization. I'm going to talk about the next couple slides here how the circulatory system is improved. And that's because we're going to extend a septum, which is a wall that separates ventricles and atria in the heart. And it's going to completely divide it into 4 chambers. But this is slightly incorrect. Your book says, it's completely divided, but there are actually some orders within reptiles where it's not completely divided. It's like 80% divided. So that's why I'm going to show you a couple slides, because I think it's fascinating how in some species it's not completely divided, yet they're categorized as reptiles. So put on the nerd glasses and go through those. No, not to my knowledge. So. Okay, so before we talk about the reptile heart, let's just talk about the amphibian heart, because I didn't really get a chance to do that. So this is an amphibian heart, and I know that because it's got 3 chambers. We have a right and a left atrium, and then we have down here a ventricle. Now, this is where blood mixes. So blood will enter in the right side and then get pumped to the lungs where it picks up oxygen, and it'll come back on the left side, and then travel down to the ventricle and then out to the rest of the body. So you can see here where red and blue mix. Well, I'll just pick it up from here. So.